welcome to the next lecture of ZOO3649, Evolutionary Genetics. This is lecture 3. We're going to talk about bioinformatics in this lecture, and basically how we can analyze all of the data that NGS is providing for us. Okay, and so let's just take a step back and ask, what is bioinformatics? Okay, bioinformatics can be defined as a bunch of tools, programs, or algorithms that we need to handle large and complex biological information. So it's a scientific discipline that is created from the interaction between biology and computer science. So the NCBI, which is the American National Center for Biotechnology, defines bioinformatics as the field of science in which biology, computer science, and information technology merge into a single discipline. And it's not just DNA sequencing data. Okay, that's the main source of this complex biological information here, DNA sequencing data. But there's also proteome data, there's RNA data, there's all kinds of big data, which we are calling omics data. Okay, and it comes from genomics, proteomics, transcriptomics, and that's why we call it omics data. And this is the data that we in particular are dealing with. So why do we need it? I think most of you realize now why we need bioinformatics, right? The first high throughput genomics technology was automated DNA sequencing, which was basically an automated way of doing Sanger sequencing. In '95, Fenter and Smith used whole genome shotgun sequencing to sequence the entire genome of Mycoplasma and Haemophilus. These are two different kinds of bacteria. And remember, bacteria have small genomes, right? So in 1995, they managed to sequence these whole small genomes. Our genomes as humans are much bigger, right? So by 1995, we did not have a human genome, okay? So in 1999, Celera Genomics, this is Fenter here, Celera Genomics, who's Fenter's company, sequenced the Drosophila genome, which is about 1 million, or probably less than 1 million base pairs, okay? And finally, the human genome of Fenter himself, a 3 billion base pair human genome sequence was generated by the Human Genome Project and Celera Genomics. Finally, so there's two characteristics of in next generation sequencing that has led to this explosion of bioinformatics in our field. The first is the high throughput, right? So NGS can generate huge amounts of data in a short space of time compared to any other sequencing technology that we have known in the past. Okay? And as I told you in the last lecture, with new long read sequencing like PacBio sequencing, this technology, by the time you are doing research, is going to be out of this roof in terms of data and in terms of things you can do with the data. So a very interesting time, guys, to be biologists. Okay, unfortunately, most biologists, including most of you and I, when I was your age, had no way of handling this kind of data. We are not used to this. Computer science guys are used to that. We are not used to handling massive amounts of data as biologists. Okay, so, and the software for handling the data are still just being written. Okay, they are getting better and better and better. Since the 5 years I've been at Univen, the software for handling bioinformatics data has become exceptionally good very quickly, 5 years only. So all of these things are improving. Okay, but one thing that is also improving, that needs to improve more, is the ability of our Bachelor of Science students to do honors projects. They need to know some programming skills. They need to know some scripting skills. Those are things that we biologists, we never needed to know in the past. Okay? When I became a biologist, I didn't need to know any of that. But that's what progress is all about. Now, to be a biologist, you have to know about those things. All my postgrads, honors, and masters sitting in my lab in FF029, they're all doing that. So that is what is required from a biology student these days. The other thing about NGS is this Illumina problem we have, right? Generating billions of small, but very small, tiny fragments, okay? And what do we have when we have tiny fragments? We need to put them back together into the right order. So we need to assemble it. And the last lecture, we've gone now through how to assemble a whole genome, a reference genome. So you know we've been through it before. If you don't know, it's because you've skipped last lecture. Go back and listen to the last lecture. We know that we need small, medium, and big pieces, for example. We have to do it in a hierarchical way. We know this already. But I put this up to show you that assembling a whole genome from de novo, from scratch, is very difficult. You know who this guy is here, right? You know him. You've seen him before. He is an egg, right? His name is, that's right, his name is Humpty Dumpty, right? So what happened to him? He was sitting on a wall. What happened? He had a great fall. What happened? He broke into many, many pieces. And what happens when an egg breaks into many pieces? It breaks into many small pieces. What do you have to do? You have to assemble him back together again. All the king's horses and all the king's men could not put Humpty Dumpty together again. Today, my master students, they don't need horses or kings or anything like that. They are putting whole genomes together again. Assembling whole genomes, mapping whole genomes. Much more complicated than putting just one egg together again. That is what our biology students have to do in 2020. Okay? All the other stuff, that was from the past. Okay? So if you are interested in postgrad, you have to become a computer person, and a big time computer person. All right? And here again, you see a whole genome being assembled. And what are these? These are all the billions of small fragments that are being guided by the bigger fragments. And you see at that position there on the genome, you go down, the coverage is huge. I'm not going to count those like I did in the last, foolish enough, in the last lecture. I'm not going to count those, but you can see this is a very high coverage genome. Okay? And this green here tells you how high the coverage is. When it goes down like this, it means it's very high. And it goes up like this, it means it's low. And you can see there, the coverage there for that position on the genome is not very high. Okay? And so this is what we mean by coverage. And so this is what we also mean by, when a high coverage area, we are able to call a heterozygote. But in a low coverage area, it is much more difficult to call a heterozygote because you're unsure what the second allele is. You know which one came from one parent. You just need to know which one came from the other. And for that, you need high coverage. Guys, you're going to be examined on that idea. You better know it. So, we use pipelines. If you can talk to one of my students, they will tell you about pipelines. Their whole lives revolve around pipelines. So what is a pipeline? It is basically a number of steps that you take to analyze your data. Now, it can be a simple pipeline that says, okay, you start with what the Illumina machine throws out, that is your raw data of DNA sequences. You go through a processing step, it gives you an intermediate result. This result, you can't actually do anything with it at that moment. You have to process it further step. Then you get another intermediate result, which you can't actually use. You have to process it in another step. You have to take the data through the entire pipeline to get a result. Okay? And your masters or your honors can be following this pipeline. And at every step you will have a mistake, you will have a problem, and you will need to spend weeks, if not months, figuring out what's the problem. Then you get to this step, you think you're a hero, but then you get another problem on this side. You spend weeks or months figuring that out, you think you're a hero, you get this result, then at the next step you get another problem. And that is what doing a master's or an honors or a PhD in bioinformatics or evolutionary biology is all about. It's about getting your data through this analysis pipeline so that you can get a result at the end. This is a simple way to look at a pipeline. What about this pipeline? Okay, now this has lots of different arrows, lots of different thises and that's. Here is another pipeline. Okay, this one is broken into 3 phases. Okay, and so what do these phases actually do? You see, you start here, you go down, you start at the top, you go down, you start at the top, and you go down, and that's your final result there. So phase 1, you process the data. Here you get your input data from your Illumina, you map them, you align them, you remove the duplicates, you look for the quality of the bases, you make sure all your bases are in good order. So you just basically aligning and checking the quality here of your data. What happens in this phase 2 of the pipeline? You now have your data clean, high quality. You take it up here and you want to say, okay, fine. I want to call SNPs. I want to find how many SNPs do I have in my data set. That means I've got several genomes, they've already been cleaned and we've taken them to the processing, and now we want to see how many SNPs are there, how much polymorphism is there. Okay, you can figure that out going through this pipeline. You can figure out structural variants, you can figure out raw variants, and then you can put the stuff, the results you get from this pipeline into the next pipeline. Okay, and this is a very simplified pipeline to show many different complex things. But you see basically what a pipeline is all about, okay? You start with the raw reads and you end up with a result. And your job as a student is to take your data through these different pipelines successfully and get an end result, okay? So there are different pipelines for assembly. I have been through the assembly pipeline with you in the last lecture. You only need to do an assembly if no genome exists already. Okay, that means you must create a reference genome de novo from scratch. Okay, and this can be useful for whole genome analysis, transcriptome analysis, ascertainment, bias variation, etc. Single variant detection, SNP variant detection. You just need to know that there are 2 pipelines for whole genomes. The first one is the assembly pipeline. Here we use the small reads, the big reads, and the medium reads and the big reads to help assemble a genome de novo, right? Once we have that genome, we don't need to do that again for a species, okay? Once we have the first lion reference genome, we don't need to do a reference genome for a lion anymore. Once we have the first mosquito reference genome, we don't need to do a second one with the second mosquito genome. We can just map it, okay? Here, with the second and third and all the other genomes, we do mapping. In other words, we resequence, okay? We resequence it and map it to the known genome. And which one is the known genome? This one that we assembled over here, okay? So here you see, you sequence raw reads from the Illumina and you assemble it de novo into different contigs, and each contig makes up the whole genome from nothing, de novo. Here you send the reads and you map them against what? A reference that is already there. This reference that you made here, this is the reference now you are using to map reads onto the second genome onto. You don't need to do the second genome from scratch. You already have the first genome as a reference, okay? So, as of 2013, this is quite outdated now, but as of 2013, already the genomes of so many organisms have been sequenced. Thousands of bacteria, by now, thousands of all of these things that you see here, okay? Thousands, if not hundreds of thousands have been sequenced, okay? Massive amounts of data. So with that, I will leave it to the second lecture, where we will get much more into bioinformatics, into more of the nitty-gritties of what it means and what bioinformatic data can do for us. Okay, so we will see you in the next lecture.